Hello, my name is Shelby Bradford, and I'm a graduate student at West Virginia University. I study how one messenger molecule in our immune system affects how a group of immune cells called dendritic cells respond to vaccines to learn how we can make these more effective in newborns. Dendritic cells are like sentinels, patrolling the body to look for things that don't belong. Their claim to fame is that they activate the adaptive arm of the immune system, the side that has to be trained to a specific pathogen but provides long-term protection. Dendritic cells are usually said to sound the alarm when something isn't right, but they also receive and respond to signals from other cells. These signals are in the form of small protein molecules called cytokines. There are a lot of these, some activating cells or turning them on, others pumping the brakes on some function or activity, and still more that say move, divide, or make this protein. My interest is in interleukin 27 or IL-27, a cytokine responsible for opposing inflammation. Most of us think of that as fever or swelling, but in the immune system, inflammation is important for recruiting cells and for activating them. I care about this cytokine because infants, specifically newborns, have a lot more IL-27 circulating in their system than do older children or adults. And this is important because a suppressive cytokine at this age prevents the immune system from reacting to all of the new things it's coming into contact with. However, it becomes counterproductive when the infant is exposed to either an infectious agent or during vaccination, when inflammation is needed to stimulate an immune response to create protection. Therefore, my interest is to understand how IL-27 affects the activity of dendritic cells during vaccination to provide insight into a novel mechanism to improve immunization for this age group. My model uses an existing early life vaccine called Bacillus Calmette-Guerin, or BCG. BCG is used to protect against the disease tuberculosis. It's a live attenuated vaccine, meaning it consists of living but weakened bacteria and is usually given at the time of birth or shortly thereafter in areas where tuberculosis is common. The vaccine has a wide range of efficacy, though, meaning that sometimes it creates a good response and sometimes it doesn't. My hypothesis is that increased levels of IL-27 at the time of vaccination contribute to reduced immune activation, specifically through a decrease in dendritic cell activation and activity. I anticipate IL-27 will reduce the ability of dendritic cells to internalize and degrade bacteria and oppose the cell's activity in presenting this degraded material, or antigen, to the adaptive immune system and producing inflammatory cytokines. These effects in combination result in a reduced immune response, contributing to worse long-term protection.